So here at the Live Lab we have a number of diverse interests, as Rami mentioned, and one of my main research interests is how we hear and understand speech and how we perceive music and what goes wrong with hearing loss. Now, how the ear and brain work together to understand speech in difficult listening conditions is quite amazing. We can understand speech with people speaking with different accents, different background noise, reverberation, that there might be in an echoey space. We can switch our attention in a group when different people are speaking, and that works very well with the normal ear. When there's damage to cells in the ear that cause hearing loss, there's big problems and a need for hearing aids. Music, in some regards, is even more difficult. The range of frequencies over which musical sounds are created is typically broader than that for speech. The range of loudnesses from the quietest to the loudest sound is typically greater in music. The rate at which acoustic events occur is greater in music. When we speak, we're limited how quickly we can move our vocal tract and our vocal cords to change the way that we're producing sounds, whereas with particularly multiple instruments as we have tonight, there's lots of events happening very quickly over a range of frequencies, and there's lots of things happening spatially at once that we want to attend to. And again, if our ears are functioning well, then we can make sense of and enjoy quite complicated music. But if we have damage to the cells in our ears that are involved in turning those sound waves into brain activity, then we can miss out on not just understanding speech, but also a lot of the enjoying music. So I'm going to give a bit of a demonstration. As mentioned, KEMA has been sitting in the audience. So KEMA stands for Knowles Electronics Mannequin for Acoustic Research. So KEMA's head has, where our inner ears would be, has microphones that we can record basically what would be arriving at your eardrums as you're listening here, and then we can use that in research to understand for real sounds being created in this type of concert, what type of sounds are being produced. We can put hearing aids on KEMA's ears and see how the hearing aids are modifying those sounds. And we can then do further work to understand how the ear and brain are processing that. So I'm going to talk a little bit about that, but first I'd like to give a demonstration with a recording that was done on KEMA in the first set tonight. So I'm going to play back an excerpt from what we just heard before, and so this is recorded through KEMA's ears, and it's going to be playing what would be heard by a normal hearing person. So if Ronald can bring that up. We can see that there's lots happening. Now go through a sequence of simulating different levels of hearing loss, from just a mild hearing loss to a moderate hearing loss to a more severe hearing loss, and then I'll go in and play with these manual controls and give it quite a severe hearing loss. So just listen to how this affects your perception of the sound. becomes more muffled. You're missing a lot of the high frequency information that the normal ear is able to convey, and there's less enjoyment of the whole range of different sounds that are being created, the dynamic interaction between the musicians. We just hear some bass from the piano mainly and bass from the drums, and everything's a little bit muddled. Now from this demonstration you might think, well, couldn't we just amplify the high frequency sounds, make them louder again for somebody who has this kind of hearing loss? One of the ways that the ear is able to do, when it's functioning normally, such an amazing job of picking sounds out of background noise and helping us to understand very complicated sounds like speech or music, is that it's not just putting together all the different frequencies that we're listening to and conveying them to the brain. It's always analyzing what we're hearing in the context of all those frequencies at once and what has come previously. So we refer to this as nonlinear and time-varying processing by the ear. So how this happens is the sound gets funneled into our ear canal, gets conveyed by the bones of the middle ear through to the cochlea in the inner ear. All of that is a linear process, so that's conveying all the frequencies through quite faithfully to the inner ear. But within the inner ear here, at any one point along the cochlea, there are some different cell types here that are involved in turning those sound vibrations into brain activity. So these pink shaded cells here are referred to as inner hair cells, and their main job is to detect these vibrations from the sound waves, turn them into neural activity that the auditory nerve fibers here send off to the brain. These green cells here are referred to as outer hair cells, and they're actually detecting the vibration, and they're boosting small vibrations in some contexts, and as we go to larger and larger vibrations, they boost them less and less, but they do this in a competitive way along the length of the cochlea, such that some frequencies get through, some get boosted, and others don't get boosted. And so it's always in the context of all the frequencies we're hearing at once, the pattern of vibration that actually gets conveyed through to the brain. Now, the way this analysis of the frequencies work and is modified by these outer hair cells is because along the length of the cochlea, from the base here up to the apex of this snail shell structure here, is that there's a membrane that at the base likes to vibrate in response to higher frequencies, and as you go up towards the apex, it prefers to vibrate in response to lower frequencies. So it's doing an analysis of all the different frequency components of what's getting into your ear and responding at different points. So these cells here are sitting along on top of the basilar membrane that's illustrated here in the middle, and so at different places along the length of the cochlea, we're getting an analysis of the different frequency components, but as I mentioned, in this non-linear way. The bad thing is that these very delicate cells are easily damaged by exposure to loud sound, by aging, by some chemicals and drugs, by some genetic mutations that can cause what we refer to as hearing loss. So what we get is this very intricate analysis of sound that falls apart somewhat when we have damage to the cells that causes then problems with understanding speech, with appreciating music. And because of this way, non-linear time-varying way that they're doing it, it's not just a matter of making sounds louder. Some of you, if you suffer from hearing loss yourself or know somebody who wears hearing aids, they might tell you, oh, when I use my hearing aid, I can hear things, but I don't understand them, or I hear them and they sound muddled. That's because what the hearing aid's doing is typically not fully compensating for the type of damage that we have in the inner ear. So what we do in my research group is develop computer models of this whole process, from the sound vibrations at the eardrum through to the neural activity. So we can take any arbitrary sound, whether it's speech or music, and play it into this computer model, and we have sections that describe what the bones of the middle ear are doing, what the filtering, frequency, filtering properties of the basilar membrane are doing, how these outer hair cells are controlling the vibration, how the inner hair cells are converting those vibrations into neural activity, and we get a prediction then of the spikes or the neural activity that's being sent off to the brain. So one thing that we've looked at with this model is, in a collaboration with Laurel Trainer, who's the director of the Live Lab, is some very basic music perception phenomena that in Laurel's lab they've studied both through behavioral experiments and also EEG to look at how the brain is responding to these. So in the first study that's illustrated at the top here, we were interested in why is it that in choral music that the soprano gets to be the diva, that the melody is normally in the soprano line in choral music, or in piano music, it would be the highest note that is normally conveying the melody. Why does this higher pitch win? And through this experiment, where we looked at pairs of simultaneous piano tones and looked at changing either the higher tone or the lower tone from the standard, and looking at brain responses to those, we found that our brains actually do respond more to changes to the higher tone than they do to the lower tone. And then we're able to, with the computer model, analyze the neural activity that's predicted from that model, and we found that it's actually this nonlinear processing controlled by the normal outer hair cells that is producing this phenomena. So this basic percept that higher pitched sounds are more salient than lower pitched sounds when played together, our ears are already creating that. If we have hearing loss and those outer hair cells are damaged, that phenomenon is going to go away, and now the sounds are going to get muddled together. It's going to be harder for somebody with hearing loss to hear out the melody in choral music or in a piano piece or orchestral music where a higher pitched instrument is conveying the melody. In a subsequent study, we looked at rhythm. We said, if we've got these simultaneous tones that are being played, and now instead of changing the pitch, occasionally one of them is introduced earlier than is expected based on the beat that's been set up. Is it changing the higher pitch that has a bigger effect, or changing the lower pitch that has a bigger effect on that perception of beat? And here again, we used brain measurements with EEG, as well as a perceptual experiment, having the subjects tap along and see how that gets upset when a note comes earlier. And in this case, we found that actually the bass note coming earlier had a big effect than the higher note. So that might explain why in music often it's bass instruments that are conveying the beat or the rhythm rather than the higher pitched instruments. Now, when we look at the neural model, we can see some aspects where already in the ear this bias towards lower tones conveying the beat or the rhythm is set up. But there's also some evidence that the brain doesn't always favor the low tone. There are contexts where they're equal or where the high tone might win out. And this is important, particularly if you think about rhythm or beat, that it has to do with expectation. Our brain doesn't know what beat the music's going to go at until we hear it. And so our brain is analyzing the statistics of what we hear and is coming up with expectations. Now, in more conventional music, where you may have musicians who are playing from a score or from memory with a more regular beat, you quickly build up an expectation of what the sound should be like, and the musicians then can play around with that and build up your expectations. They can violate those expectations, and it can be a surprising element to have through improvisation or through a classical musician playing around with the tempo to slightly violate what you're expecting. Now, the type of improvisational music that we're hearing tonight, they're starting out with trying to really stretch our brains and have our brains have to work very hard often to build up a representation of what we're hearing and what we're expecting, and more often than not we're getting a violation of what we might be expecting that in many ways can really stimulate our brains and give a very interesting listening experience. Again, that's happening if we have normal hearing, but what's happening in that case if we have hearing loss? So I'm showing, just to finish up, some illustrations that I've created now with an excerpt that from last night's concert that I had time to run through my computer model to give you an illustration of what the brain's getting in the case where we have normal hearing, where we have hearing loss, but no hearing aid, and finally when we have a hearing aid that's trying to compensate for the hearing loss. So this is the short excerpt. This one's only 3 seconds long. Okay, so a short excerpt there, 3 seconds long. The top plot here is showing what's referred to as a spectrogram. So this is showing us a function of sound frequencies over time, how much energy there is acoustically at those different frequencies. So where it's blue, there's not much energy. Where it's the light blue to yellow, there's more energy. Where it's green going through to red, there's a lot more energy. And you can see there's a fair amount happening there. In the bottom plot, now we have the prediction of how the auditory nerve is conveying information to the brain about those acoustic responses. So now we have, we're modeling auditory nerve fibers that are tuned from high frequencies in the base of the cochlea to low frequencies up in the apex of the cochlea over time. And now the color is showing how many neural discharges or spikes are being sent off to the brain in response to these sounds. And you can see that there are interesting patterns of red events here that are occurring that are conveying these musical notes and percussive sounds in this short excerpt here. So that's what we get for the normal hearing case. If we were to introduce a high-frequency hearing loss, so a bit of hearing loss at the low frequencies, but more and more as we go up to high frequencies, and provide no amplification, so no hearing aid provided, this is what the neural response would look like. So again, the spectrogram hasn't changed because we haven't amplified the sound, the acoustic signal's the same, but you can see now that the neural response here is mostly missing neural activity because the hearing loss means the information's not getting through in those high frequencies, and it's very weak here at the lower frequencies. There's a degraded representation here of the events that we saw at the lower frequencies here in the normal case. Now, what about applying a hearing aid? So now I've got the same hearing impairment, and now I'm looking at providing amplification that compensates fully for that amount of hearing loss. So where there's a little bit of hearing loss, a little bit of amplification at the low frequencies, at higher frequencies where there's a lot of hearing loss, more amplification. Let's look at the neural response, or the predicted neural response, and see how the model does. So first, if we look at the spectrogram, we can see the effect of all this amplification. We've got a lot more high frequency energy now from the acoustic signal. If we look down at the predicted neural response here, we see we get back more high frequency response here. There's a stronger low frequency response, but if we compare the left and the right, the normal and the impaired case, we see that the amplified hearing impaired case doesn't exactly match the normal case. There's a lot more spread across different frequencies here in response to each of these sounds, and even more so at the high frequencies. We've got lots of auditory nerve fibers responding to the same acoustic event, whereas they're much more discrete in the normal case. So that's, again, consistent with what people with hearing loss and hearing aids say, is that they can hear something, but it's very garbled or muffled. It's not distinct like normal hearing is. So we've got a long way to go in improving hearing aids, both for understanding speech, but even further for music. So how can we do that? So one of the tools that we have is KEMA, and even more so, the Live Lab, a place where we can create live music, we can play around with the acoustics, we can take measurements with KEMA, with and without hearing aids, to do real tests of what the hearing aids are doing. But then also we can bring in audience members wearing hearing aids and test their perception through the audience physiology. We can test how their brains, their heart rate, and so on are responding to their listening to music. And our aim through that is to improve listening to live music for many people who otherwise would love to enjoy music but are suffering from hearing loss. So I hope that's given you a little bit of insight to some of the research that's going on here at the Live Lab and the ways that we're trying to connect in helping the society, helping people with hearing loss to be able to appreciate music again. And with that, I'd like to invite CCMC back for the second set. Thank you.